بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. So I would like to present my recent research in Lund University in regard to touch response implants. So before I start, I plant during its growth, it's faced and encountered by many obstacles that affect the productivity and the yield, and also the growth. So we have biotic stress and abiotic stress, and a lot of studies done before for the effect of this biotic and abiotic stress on plant. So this new research topic that I started when I started my postdoc in Lund University, we started to see how plant, if the plant is responded to mechanical stress, like touch, like wind. So mechanical stress come from if one insect landing in one leaf, like drops of water, wind, agricultural practice, and ruling. So all these practice can lead to mechanical stress on plant. So the question is this mechanical stress is harmful for plant or useful for the plant? So when plant exposed to mechanical stress, it start to have thigmomorphogenesis. It means that there is a change in morphological of plants. So we have increase in root systems, we have shortened stem, we have more branching, and the most important thing and interesting thing that we have more tolerant to some abiotic stress, and the abiotic stress like for drought, lodging, pathogen. And this treatment by mechanical stress increase the tolerance of plant to biotic and abiotic stress. So how this mechanical signaling come from? It's come from touching, brushing, pressing, bending, wind, sound. And all these factors affect the plant and cause mechanical stress. So it's used for improved yield of plants, also improve quality of plants, and for improved tolerance to biotic and abiotic stress. So it means that with only eco-friendly treatment, without any using chemicals, I can improve the plant in friendly way, eco-friendly way. So here, if you have a look to these Arabidopsis plants growing, and this untouched, and I keep touch the plant, 2-week-old plant for 2 weeks, and this is the result. So I have delay in flowering because this stem inflores, and you see that there is a reduction in rosette size in touched plant. It means that touch has effect on flowering time and also in rosette size. I did this recently with this one gene that controlling the signal cascade. So when you look out this gene, you see this plant become less sensitive to touch. And we use this gene to identify the signal cascade in touch response. So I would like to present this interesting practice that long time ago in Japan, they used to improve winter wheat. So what they do, after sowing wheat during November, 3 weeks old plants, they start to stepping on the plants. And this treatment can improve the yield around 20 to 30% in improving the yield. So you can imagine that how this treatment can affect the yield. So not only stepping or pressing the plant, they also use like ruler, metal ruler pulled by tractor. And this can lead also to the two strategy to apply mechanical stress on plant. In the lab, how can I apply this one? Do I have a different way to apply this one? By like curvature of the plant, all speaker, like sound. So I can use this application like a sonicator. All this application makes the plant to consider mechanical stress on plant. So we need, if you have a look to this table, I think it's not clear, but I will summarize. So we apply mechanical stress in different crops like maize and different plants like potato, bean, wheat, and we use different mechanical method. Some plants treated with bending, brushing, or wind, and the result that these treatment increase the tolerance of plants to biotic and abiotic stress. For instance, this increase drought tolerance. This treatment increase drought tolerance in bean. So it means that there is good effect of mechanical stress on plant. So in the lab where I work, we publish a paper here that show that there is two ways or two pathways to control touching implant. One is depend on jasmonic acid, and one is depend on jasmonic acid independent pathway. You see this figure? When plant received the mechanical stress, there is a change in microstructure of cell wall and plasma membrane. And in plasma membrane, there is some channels called stretched activated channels. When the membrane is affected by mechanical stress, there is some stretching to this plasma membrane, and this leads to activate this channel and influx of calcium inside the cell. And influxing the calcium inside the cell lead to spike in calcium and lead to activation of downstream signal cascade. And this lead to two pathways. One is dependent of jasmonic acid, as I said, and one is dependent jasmonic acid independent pathway. And now we can identify these two important pathways in response to mechanical stress. So the result, impact on architecture, flowering, biomass, and defense. So there is no study before showing how cereal respond to mechanical stress. There is some studies before shows that when plant exposed to mechanical stress, there is change in composition of lignin in cell wall. But there is not accurate study how cereal respond to mechanical stress in transcriptomic level. So we start this experiment to see how these cereals respond to mechanical stress on transcription level. So we have transcriptomal analysis of touch response in cereal crops, barley, wheat, and oat. So I will not talk about this, the importance of these three crops, because my colleagues before, and also you know how important these crops for feeding, for human consumption, and for fodder and for beer, for like barley especially, and oat for human consumption, and wheat for also for human consumption. So it's very important, this plant for human. So how we design the experiment? So we have 2-week-old barley, oat, wheat plants, and then we treat this plant by mechanical stress. How I treat them with mechanical stress? By brushing the leaves. So I will present now. So this is what I did. I have brushed 6 centimeters, and then I brush the whole seedling down up for 10 times. And you can imagine that only this touching can upregulated thousands of genes in these 3 species. So this first result, PCA plot of gene expression. I have 3 replicates, and here is nicely see that these different time points is tightly separated. It means that there is a clear effect of touch. Also here, we have clear separation between these time points. These consider one time point, 3 replicates, and these 3 replicates, and these 3 replicates. So it means there is a nice separation and tight separation between these 3 touch or time points. So here, if you see, when I treat the seedling, what is the result? You see here, there is 25, after 25 minutes from mechanical stress, I see many genes upregulating, because red columns consider upregulation genes. So you see that upregulation genes is higher or more than downregulation regulated genes. Also, it was interesting that the highest number of differentially expressed genes was found after 25 minutes. It shows how fast the gene expression in response to touch in the 3 serial. After 25 minutes, I found the highest number of differentially expressed after 25 minutes. Also, it's worth mentioning here that here I have 2,000 genes of oat induced after 25 minutes, but after 1 hour, it's sharp decline again from 2,000 to 828. It means that oat has more transit expression than barley and wheat. It means that wheat and barley have a stable expression more than oat. And if you see this diagram, Venn diagram, it shows that overlapping between the different time point in barley. So this means that this all genes is expressed after 25 minutes, this after 1 hour, and this after 2 hours. So we need to check what is the overlapping between these 3 in the genes in the 3 time points. And if you see that 25 minutes and 2 hours have 60 to 75% unique genes and not sharing with other time points. But for 1 hour, we see that there is about 60% sharing with 25 minutes and 2 hours. So to obtain overview of the different gene expression pattern after touching in serial, we have pattern. It means that how genes looks like over time. So I classify this pattern to different category. So we have early up transient cluster, mid up transit, and mid up transit. What does it mean? This category or patterns that upregulated after 25 minutes and then return to the basal level after 1 hour. And mid, it means that it reached its maximum. So this gene reached its maximum expression after 25 and 1 hour, after 1 hour, and then return to basal life. And we see that this category is considered the highest number of genes, contains the highest number of genes. It means that the pattern of expression in serial for upregulation gene, that after 25 minutes, it's go up and reach its peak, and then to go down to the basal level. There is another interesting pattern. The genes start to increase until it reaches its peak after 2 hours. So if we talk about the pattern for down-regulated genes, you see here also the same. This pattern shows that genes go down after 25 minutes and then go up to basal level again, or continue upregulated over the time course. And interestingly, I forget to say here that this category of genes, when we check what these genes do in this pattern, and you found that it's really represent the components and the genes, main genes in touch signaling pathway. And this fit with what we found before in Arabidopsis plants, that this components is related to touch response in plant. For down-regulated gene, it was lignin and secondary metabolites is affected and is representative for down-regulated genes. So now we try if all the previous study was done on Arabidopsis plant, this dicot, and this is the first time to study monocots in cereals. Monocots is as study cereals. So is there any pathway common between dicots and monocots in response to touch? To answer this question, we did what called orthogroups, and we have orthogroups here, and we have overlapping between different time points, like 25 minutes. We have the orthogroup of barley, the orthogroup of oat and wheat, and Arabidopsis. So we found that there is a common orthogroup between Arabidopsis and the three cereals in different time points. And there is a good thing that we found a unique orthogroup only found in cereal. It means only found in the three species. So to dig in and to study what's common in this common four, we found that it's in share in the base, like classic touch responsive genes, MAP kinase, jasmonic biosynthesis, and calcium signaling and transcription factor. But we still can identify a new components only found in cereal, and this consider cereal-specific genes like soprene biosynthesis, callose biosynthesis, lignin biosynthesis, and melatonin biosynthesis. So these considered only unique in cereals and not found in Arabidopsis. So to validate RNA-seq data, we did qPCR, and also we increased the time course because before in RNA-seq analysis, we use only 25 minutes, and we use only 25 minutes, 1 hour, 2 hours. So in this experiment, we need to increase the time points. So we add one early time point, 10 minutes, and late time points, 4 hours, to see if the plant still expresses these genes after 4 hours, and also in 10 minutes. In the same time, we need to study if we touch or brush this leaf locally, we call it local leaf. Is the signal well transmitted from this local leaf that we touch to the neighbor leaf or not? Because they are vascularly connected. So we expect that if we touch this leaf, there is a signal that will transmit it to the systemic leaf, and we need to check this the expression of genes here. Bunch of genes, all touch responsive genes, and these genes induced by touching, and we can identify these genes. So we check these genes, the expression of these genes, and the interesting you found here oat, barley, and wheat. And this blue line shows that there is a significant increase after 10 minutes of touch treatment. Sorry for that. Yeah, so there is, you see here, there is increase in expression of this gene. This gene considered be touch responsive genes. And for the same for barley and for wheat. The conclusion here that you see that here in barley and wheat, the expression still or remain after 4 hours, and still significantly in comparison to 0 minutes. But for oat, the maximum increase happened after 25 minutes. And the red line considers the expression in the systemic leaves. And we can still see there is expression, increase in accumulation of transcript in this gene after 10 minutes and 25 minutes. It means that touch signaling can transmit from the local leaves to another leaf, that neighboring leaf. So, as you remember in my first slide that I told you that touch signaling in plant is controlled by two pathways, jasmonic acid pathway and jasmonic acid in the dependent pathway and independent pathway. That response of plant to biotic and abiotic stress. So in the two pathways that I showed before, one is related to jasmonic acid and one is independent. This study was done in Arabidopsis. So the question was, is these two pathways still exist in cereal? And to answer this question, we apply methyl jasmonate treatment, volatile, and we check if there is increase in expression of touch responsive genes in response to only jasmonic acid or not. Use volatile methyl jasmonate to avoid physical contact with the plants. And interestingly, we have found some gene that upregulated after 2 hours. So you see here, control 2 hours. It means that plants that contain only air, can subjected to jasmonic jasmonate. Can clear there is after 2 hours, and there is significant increase after 8 hours. So it means these plants respond and respond to touch. That there is a for jasmonic acid dependent induction. Interestingly, that other pathway, jasmonic acid induction, we can prove this way cannot find increase after exposure the plant just so control 2 hours, and this 2 hours no significant. So we have a proof cereal has also pathway, jasmonic acid independent pathway and jasmonic acid independent pathway. Also, there is accumulation of jasmonic acid after 25 minutes. And this form of jasmonic acid called jasmonic acid isoleucine is the active form of jasmonic acid. For jasmonic acid, we cannot see itself accumulation, but we found it's in the active form of jasmonic acid, which considered jasmonic acid isoleucine. So conclusion, cereal respond to mechanostimulation with similar early induction as involved in Arabidopsis, that observed in Arabidopsis. It means that we almost see that after 20 minutes in Arabidopsis, we have accumulation of transcript of touch responsive expression, and the same happen also in cereal. It means that they both respond early as each other. Second, we can identify cereal specific responsive gene. Does not exist in Arabidopsis. Also, jasmonic acid and jasmonic acid independent and dependent signaling pathway operate during touch response in cereal. Oat and barley appear to have a stronger systemic response to touch than wheat. Change in cell wall genes after mechanostimulation are very prominent and show interesting pattern. The response in oat seems to be shorter than wheat and barley. So I'd like to thank my colleague in Lund University. Yeah, Olivier Van Aken is the head of the group, and Kasim and Kong and Yula and Abraham is like doctor and master student. And this group is collaborate in bioinformatics, and they collaborate to analyze RNA-seq data. Okay, and thank you for listening.
Thank you very much for the time now for
have you linked the response? Have you linked the response to mechanical whatever to specific receptors on the cell surface?
Okay, so, yeah, thanks for this nice and very good question. So till now, you know that the receptors of mechanical stress exist on the plasma membrane, and there is two types of channels, like it's called stretch activated mechanochannel, and there is mechanosensitive calcium channels. And we check the mutants of these receptors, but we can these mutants still work as like type, wild type. It means that they are not involved totally in touch response. Because if we would like to know the role of these receptors, so we knock out the gene that express these mechanochannels, but we cannot identify exactly which receptor play an important role of perceiving the signal from in plasma membrane and transmit it to inside cell. So we still work in the downstream effect, like downstream transcription factor, but this upstream signaling, like receptors, we have not identified it. Is this your question?
The receptors are affecting the outside effector on the side of the cell.
Yeah. So we know that there is maybe potential mechanosensitive receptors, but till now, after knock down these mutants, knock down these receptors, we still have wild type response. So it means that's not totally involved in touch response implant. So
the proper
yes. So we checked. There is some mechanosensitive called mechanosensitive small conductants like MSL, and there is mechanosensitive calcium channels MCA, and there is piezo is also receptors found in mammalian cell, work as a receptor for touch, and it exists also in plant. And which lately we check, we have mutant from piezo and other receptor MSL and MCA, but and they can still have normal gene expression, normal wild type response. So it have not have like direct effect in touch signal. So still have to look for the upstream signals for this touch signal. Thanks.
Further questions?
Thank you for this nice presentation. I only have just one question. You mentioned at the introduction that this mechanical treatment could increase the yield by 20 to 30%.
Yeah.
I believe that we were talking about the arabidopsis, right?
No.
You were talking about the ceiling.
Yes. So what I explained as first of my talk, there is practice long time ago, since 18th century, that practice in Japan. So what they do, after sowing the winter wheat in November, after 3 weeks from sowing the winter wheat, they start to stepping on plants. And this practice called mugi-fumi, and this one can search. So I found that there is a paper there, and I think I can give you the reference for that. So it's Linda 2014. Yeah, so it's LIDA 2014. And they can, by practicing on stepping on plant, they have increased yield. But the problem is, if you would like to repeat this experiment and have the same results, it's not easy. From land to land, the moisture content of the soil, the temperature, the humidity. So it's not easy reproducible, because we try once, we get some effect, but not this increase in yield. But we have like different effect, like we have some increase or decrease in the plant height and yield, but it's not significant. So it means that we should be repeated more times to prove this. But this proven practice in Japan since like long time ago, and it's published that they increase like 30% in yield of this stressed plant.
But under your, for your research,
Yeah,
how
for
you evaluated this,
I tried it once. Yeah, so it's give me like little bit increase in yield, but we still need to consider a lot of difference in condition in soil between Japan and Sweden. Welcome.
Question?
Yeah, this one. Yeah. Yeah. Yeah, so it's a good question. So when we start to make this overlapping, we make it by ortho group. So we use ortho group to overlapping between these three genes, not only the gene, the ortho group of these genes. So it means this overlapping, the ortho group overlapping, not the gene overlapping. So if we have a list of genes that over differentially expressed in barley and wheat and oat, there is ortho group belongs to these genes, and there is maybe some genes have the same ortho group. So if we calculate like if you have 10,000 genes differentially expressed, but if you check the ortho group, maybe it's 700 ortho group. So when I compare, I use ortho group to compare and overlapping between these time points. Yeah. Okay.
Another question is the gene what you try to validate.
Yeah. Yeah.
So from this data, I found that it's already known that barley is more tolerant to abiotic stress
Yeah.
than the other two genes.
Yeah.
From this data, can you confirm this?
Yes. Yeah. So it's a good point and good question. Thanks. So if you see here, when I check the touch responsive genes in the three species, it was clear here that in barley, you see the level of expression in barley very high in comparison to oat and wheat. And this maybe reflect the higher tolerance of barley in comparison to oat and wheat. So is this, I think this good point, I think it's you need to consider because it's really higher induction and transcription level is higher than both of them. And it's not only like, it's like 30%, I think. So it's like higher increase of level of expression. Yeah. So it's like higher induction than oat and wheat. No. This is, yes, this just molecule dependent and independent as well. Yeah, you're welcome.
Thank you very much.
You're welcome.